This is the final video for the cellular respiration unit. In this video, we will be discussing fermentation and an overview of other catabolic reactions. As we've discussed previously, phases 2 through 4 of cellular respiration require oxygen to occur, but glycolysis does not. This means, in the absence of oxygen, cells can still use glycolysis to obtain some ATP. This is much less efficient, though, because there are only 2 ATP produced through glycolysis. Additionally, in order to continue running, glycolysis still needs a supply of NAD+, which is no longer being produced by the electron transport chain without oxygen present. Instead, the process of fermentation is used to regenerate NAD+, to allow glycolysis to continue. There are 2 major fermentation pathways. The first is lactic acid fermentation, which occurs in animals and bacteria, and is called lactic acid fermentation because pyruvate is converted to lactic acid. While some bacteria can live only on the ATP produced through fermentation, lactic acid fermentation in animals occurs only in the muscle cells and only for short periods of time. This occurs usually during heavy aerobic activity when muscle cells may not be getting the required oxygen they need for cellular respiration, and lactic acid produced is a waste product that can harm the cells and must be removed. The second type of fermentation is ethanol fermentation, which occurs in plants and fungi. Here, pyruvate releases carbon dioxide as the 3-carbon pyruvate is converted to 2-carbon ethanol. This is the process that humans have taken advantage of for beer, wine, and liquor production. Similar to lactic acid fermentation, ethanol is a waste product for the cells, highlighting that the only purpose of fermentation is to regenerate the NAD+, to allow ATP to be continued to be made through glycolysis. A variety of carbohydrates are consumed and digested, all of which can be used to produce energy. Some disaccharides contain glucose monosaccharides as building blocks, such as the 2 monosaccharides that make up maltose, and a single monosaccharide in lactose and sucrose. Once digested, these glucose molecules will directly enter glycolysis. However, fructose and galactose will instead be converted into glycolysis intermediates, products that will come later in the pathway. For example, fructose can be phosphorylated and enter glycolysis at step 3. If you remember the function of macromolecules that you learned earlier in the course, you should know that carbohydrates are not the only energy source. Lipids called triglycerides are also a good energy source. After a meal, the small intestine absorbs triglycerides, and these are broken down inside cells to glycerol and fatty acids. Fatty acids are then broken down, releasing energy that will be used to make ATP. Fatty acids are broken down in a process called beta oxidation, which removes 2 carbon units from their ends. For example, this fatty acid has 16 carbons. And after beta oxidation, it now has 14 carbons. This does not produce any ATP, but does produce 1 molecule of NADH and 1 of FADH2, which can then travel to the electron transport chain to make ATP through oxidative phosphorylation. Additionally, the 2-carbon subunit that has broken off is acetyl-CoA, which will feed into the citric acid cycle to produce more ATP and electron carriers. A much greater amount of ATP can be made from a fatty acid molecule than a glucose molecule, which is why triglycerides function as long-term energy storage for the cell. The catabolic process of cellular respiration is key to producing ATP, which is constantly being broken down and resynthesized. The level of ATP in the cell is an indicator of how much energy a cell has available. When ATP levels are high, the cell has a large amount of free energy, which means the cell does not need to keep making ATP. In response to high levels of cyclic AMP, key enzymes of cellular respiration are inhibited or downregulated, causing less ATP to be generated. One such regulated enzyme is called phosphofructokinase 1. This is an allosteric enzyme with many activators and inhibitors. When the levels of ADP or AMP are high in the cell, these molecules will bind to PFK1, shown here, and activate the enzyme so that glycolysis continues, increasing the amount of ATP in the cell. However, when levels of ATP are high, ATP itself will bind to phosphofructokinase inhibiting the enzyme, causing glycolysis to slow down and produce less ATP. Another inhibitor of the enzyme is citrate, an intermediate of the citric acid cycle. When the cell does not require any more ATP, glucose is stored in long polysaccharides in the cell. This is an anabolic reaction that covalently binds individual glucose molecules into long branched chains. In animals, glucose storage molecule is called glycogen and is found in muscle and liver cells. In plants, the glucose molecule is starch. Once the cell needs ATP, it is able to break individual glucose molecules from these long branches and feed glucose into the glycolysis pathway.